Thank you very much. We are still keeping very well up on time with our outline schedule. And it was said just now that curiosity is very crucial when you're a researcher and when you want to change the world. It's also very important to have a vision and a dream. So thank you very much for those words. And now I'm very happy and very glad to welcome our first presentation. We see soon when she will be up. Where is she? Liat Ariel from Hebrew University.
Hi. Have you noticed this growing trend on social media where influencers proudly say that they skip sunscreen, despite decades of research proving that the sun exposure is harmful to our health? Influencers today share on social media that sunscreen is not needed. They undermine science. Their stories spread very fast. We see the same thing with vaccines denial, with conspiracies like Earth is flat, and with claims that antibiotics are dangerous, for example. Some even say that it's all a scam by pharmaceutical companies. So who should I trust? I call this the truth of lived experiences, shaped by emotions, personal beliefs, and subjective viewpoints. On social media platforms, algorithms prioritize emotional content. Algorithms prioritize emotional content at the expense of factual truth. And in this virtual spaces where communities reinforce their beliefs are called echo chambers. Now imagine a student, a teenager, who grows up in an echo chamber because he consumes most of his information from social media. By the time he reaches school or college, he already holds distorted knowledge that contradicts traditional truth. Cut between these two conflicting forms of truth, how do you think he will react? Well, research shows that existing beliefs often override factual truth. And when educational institutions try to fight misinformation by adding more facts, they end up adding more bricks to the very same wall they try to dismantle, what eventually leads to a complete rejection of science, facts, expertise, of truth. So how do we help a student grown up in an echo chamber to adapt to a world of factual scientific truth? I propose a new pedagogy grounded in philosophical and psychoanalytical theories to bridge or to recreate trust between the echo chambers and institutional knowledge. It starts by engaging students, offering them to share their subjective beliefs inside the classroom, to help them understand, first of all, ground themselves in their beliefs, and then gradually engage them with factual, objective reality. Because if we won't rethink how we teach truth, we might end up with a future generation with distorted knowledge. When in educational institution, if educational institutions won't allow space for contested truth to emerge within the classroom, then students won't be able to bring themselves to the classroom and eventually engage with objective reality. What we teach matters, but how we teach truth is really important for the future. So the next generation is listening. The question is, to whom?
Presenter Shacha Ben Itzak from Reichman University.
Good afternoon, everybody. My name is Shacha, and I'm a postdoc in Reichman University. Now, I want to begin with a true story about a wonderful person I know named Ilana. For the past few years, she's been struggling with memory decline. And in order to cope, she subscribed to an online cognitive training platform offering a variety of exercises and to improve memory and other cognitive abilities. She incorporated this into her daily routine, which was fantastic, because in 2017, the World Health Organization released a report on dementia prevention. It identified 7 key interventions that could reduce the risk of dementia by over 50%. And one of them was promotion of cognitively stimulating activities, exactly what Ilana was doing. But after a few months, Ilana came to me and said it wasn't helping. It felt repetitive and boring, and eventually, sadly, she gave up. But this wasn't the problem with this particular app. This is a broader problem with the field of cognitive training. This field has been researched for over 30 years with heated debate. Advocates and skeptics are locked in a tug of war, unable to decide if this kind of intervention is actually effective. As part of my research, I identified an overlooked factor, one that might tip the scales, offer a new path forward, and help the field finally break out of its torment. The factor was social interaction. Now, you need to understand that social interaction by itself is a form of cognitive training. It engages cognitive abilities such as working memory, inhibition, mental flexibility, and a whole lot more. In my study, I found that combining these two elements placed the trainee in a lifelike setting, thus enabling far transfer effects of training gains beyond practice tasks. But here's the true secret. In my study, for the first time, social interaction was not just added to the training. It served as the very foundation of the training itself, the means through which the training took place. Or in other words, as the Talmud teaches us, just as one iron sharpens another, so does one person sharpens another. And thank you so much for listening, and I would love to hear your questions.
Has this really never been done? This kind of social
I have
interaction?
2.5 minutes. This has never been done, not in the way I did it. The social interaction was the means to which the training took place, and I was quite aware that this question would come. I didn't just add social interaction to the cognitive training. The social interaction was the training itself. And if I would have a few more minutes, I could explain, but this is a very short time to do that.
Thank you.
Thank you so much.
Yes, now the time is up. There was a problem with the timer. Could you please make sure that... Okay, thank you. Yes, so I'm very happy to announce Ivy Batahaya. Ivy, okay. From Technion. Thank you.
Okay. Hello, everyone. Cancer is recently been recognized as more of a systemic disorder than an organ-related one, which means that biomarkers distribute throughout the body and not just specifically in the organ where the cancer first started. So why then do we not have access to fluids instead of doing solid tissue biopsies for cancer? Since we know that the biomarkers distribute throughout, it should also be obvious to us that they obviously transfer to tears, blood, sputum, breast milk, all throughout the body. But how do we access that and make biofluids a source of cancer diagnosis instead of going all the way to tissues and biopsying the tissue itself? One way to do that would be to actually probe biomarkers that are relevant and stay relevant despite being transferred throughout the body. In the blood, there are differences in pH, which then digest the proteins and DNA and RNA that are floating around in the blood. However, there are specific particles also in the blood called extracellular vesicles, which are lipid-encapsulated membranous particles, nanometers in size. Everything contained within these nanoparticles stays intact. The pH does not affect anything that remains inside enclosed within the vesicular space. The reason we cannot do this yet is because these samples are extremely heterogeneous. These particles could be 20 nanometers or 200 nanometers in size. They could be positively charged or negatively charged. They could contain whatever cargo they want to pick up from whatever organ they please. So it's really difficult to profile them one by one and really identify whether there are any changes in healthy versus unhealthy samples. Okay, so for this, in our lab, we built this nanofluidic device that fits on the tip of your finger. Here it is. And basically, what we're doing here is we input a sample at this space, bring in the labeled sample into the space mentioned between the two chambers, and then apply an electric field. Just the way that the particles move from one end of the channel to the other end of the channel will give us insight about their size, about their charge, and about their biomolecular profile. This is one really interesting way that I believe we could really make liquid biopsies more of a reality than what we have right now. Thank you very much, and I'm open to questions if you have any. Thank you.
Thank you very much, and I welcome Jonathan Eriksson from Technion.
Thank you. Hi, my name is Jonathan Eriksson. I'm really excited to be here to share with you our work on one of the largest problems in optics, which sadly is not very well known. So a lot of people, including myself until I heard about this, take glasses for granted. But unfortunately, in the developing world, over 1 billion people who need glasses don't have access to them. So try to imagine a world where you don't have glasses. You can't read, you can't drive, not to mention sit in a classroom. And the problem is how we make lenses nowadays. Basically, you take a block of glass and you cut at it and you polish it again and again until you have a nice, smooth, spherical shape. And this requires very heavy infrastructure, which is basically non-existent in the developing world. So we believe that if we change and redefine how you make lenses, we can change the problem of vision inequality in the world. In our lab, instead of polishing and cutting glass, we've developed a method to take liquids and shape them into smooth lenses. As you can see both in the bottom view and side view, we basically take a polymer, we inject it into an open ring, which is placed inside a water tank. Because of the water, the buoyancy force cancels out gravity, and in this unique case, the polymer will take a smooth spherical shape, which is basically a lens. We then solidify the polymer with UV light, and you can just put your hand inside and take out a solid lens within minutes. The water, of course, is ready to be reused for the next lens in turn. We've already created hundreds of glasses just like this, and the whole process is done in this small, portable, battery-run box, which makes it perfect for the developing world. Now, we've already contacted NGOs such as Engineers Without Borders, which are planning to take our box, take our method to schools in Ghana and start making our vision a reality. So I'm really excited to see that happen, and I'm really thankful to be here. I'd be happy to take any questions.
What's left to research?
So usually when you buy glasses, you have coverings on them, coatings, so anti-reflective and stuff like that against UV. So we're working on that. We have a cooperation with Shamil, if you know, from the north. They do their own coating. So we're trying to see if the polymers we're using can be used in the same coating method. So that's one topic which we're still working on.
Can you very briefly say what the relationship is between your work and Moran Berkovic?
He's my PI. He's the advisor. Okay. Yeah. So this Moran Berkovic, if you've mentioned him, he's the brains behind the concept, and half of our lab, me included, each one takes a section of this work, and we're working on making it better. Thank you.
Thank you very much. We are very good with time. Next one is Anna Gorevich-Shapiro from Weizmann Institute.
Doctor, will this treatment help me? My patients often ask. Well, we are now very happy to be able to cure many more patients with blood cancer than ever before We still need better tools to understand which patient would benefit from which treatment. I am studying a transformative immunotherapy called CAR T cells. CAR T cells are immune cells obtained from the patient, taught to recognize and attack cancer cells, and are infused back into the patient. CAR T cells have changed how to treat lymphoma. We can now help many more patients that do not respond to other lines of treatment. But 60% of patients will eventually relapse. Others will endure toxicities. Identifying those most likely to benefit from CAR T treatment is key to improve prognosis and reduce toxicity. So how do we know who will benefit from CAR T treatment? To break this wall, we collaborated with medical centers across Israel and the U.S. To study the immune system of patients with blood cancer. We used a high-resolution technique called single-cell multiomics to build a detailed immune atlas of the immune system in cancer patients. We then correlated the cellular populations and the gene signatures that we found with treatment outcome. We identified a specific population of cells and a genetic signature that is predictive of treatment success. To transform the biological data into a practical clinical tool, we trained a machine learning model on the biological data to predict CAR T outcome. This is the foundation for a novel blood test for a biomarker for CAR T success. As ever more patients are due to receive CAR T treatment for cancer and other autoimmune diseases, this is the first step for personalized medicine to help patients and physicians predict benefit, reduce toxicity, and guide treatment decisions so that we could always answer yes.
You get a few more seconds because of the microphone issue.
Well, I'm your questions are welcome. Okay, thank you.
Thank you, Anna. I know that your lab was destroyed and now you even got some destruction in your pitch. I'm very sorry for that. Hopefully it will not happen with the next one. I'm welcoming Yara Hamshavi from Bar Ilan University. Thank you.
Okay, so over 800 million people around the world have diabetes, and these numbers are increasing rapidly. So that means that 1 in every 10 people has diabetes. And statistically speaking, each one of us here today knows at least one person affected by this disease. So diabetes is not just a number or a diagnosis. It's a chronic condition where the pancreas cannot produce insulin, or that the body cannot respond to the insulin it produces. So the main player in this disease are the pancreatic beta cells that produce the hormone insulin that maintains normal blood glucose levels and keeps us alive. So insulin discovery was a breakthrough in medical history. Actually, 3 Nobel Prizes were awarded to people to work related to insulin, showing how much important this hormone is to human health and science. So unfortunately, nowadays, there's no cure for diabetes. Diabetic patients only supported by exogenous insulin injections, despite the fact that these injections are invasive and costly, it doesn't halt the disease from progression. So we offer the project. In this project, we offer a way to make diabetic patients insulin independent. So when we mutated a specific gene encoding for a specific biochemical pathway in chemically induced diabetic mice, we were able to see that a significant reduction in their blood glucose levels, and these mice had an insulin independent. So when we search for a compound that could inhibit this specific pathway, when we administered it to a chemically induced diabetic mice, we saw the same effect, a significant reduction in their blood glucose levels, and they were insulin independent. And when we turned to the NOD mice, a special mice model for autoimmune diabetes, so when we gave this drug inhibitor to these mice, we were able to see the same effect, a significant reduction in their blood glucose levels, and they had insulin independent. So this repeated result, we're getting the same result in multiple mice models, show us that there's a promising therapeutic component to diabetes. So I can say now that we are breaking the wall of insulin independence and diabetes, paving the way toward a long-lasting cure. Thank you.
Thank you.
Our next and last pitch will be by Ronnie Hillel from Technion, and I have to apologize. In the program, there is a mistake. Our chief designer was already not available, so I took it on me, and I was obviously too tired, so I apologize. So the title in the program is wrong. Also on the voting sheet outside, you will see the real title here, Breaking the Wall of Architecture, Bacteria, and AM. Ronnie, it's your stage.
Thank you. Hi. So what is the connection between architecture, bacteria, and robotics, or additive manufacturing? So the answer lies in Earth. Earth is local material which has proven environmental and thermal benefits. In history, it was a primary building material, and in some places it still is. So why do we still use concrete as main material for construction? Well, even though it's one of the most polluting industries in the world, causing 8% of global CO2 emissions. Because Earth has its challenges. Earth has a low strength, it's limited in geometry. Building with it is slow, labor intensive, labor intensive and not really efficient. So before I go to the solutions and innovation, I want to ask you something. Do you know the feeling when you're in your home and it's freezing during winter and super hot during summer, with the AC is still on? So that's concrete. It stores temperature, but it doesn't regulate it, and the Earth does. Earth is a natural insulating material. It regulates humidity and improves indoor air quality. So what if we could use it again? What if we could use it by moving forward and wrap our building with Earth, making them healthier and better for the planet? What if we could 3D print them from Earth? And I suggest doing that by combining architecture, biology, and robotics. First, I take natural process, which involves stimulating native bacteria to create calcium carbonate, which reinforces the soil and improves its properties. And then I integrate it with robotic 3D printing, additive manufacturing to create strong structures and complex. So we can use it as an efficient process, which I'm sorry, I'm excited. Which is more efficient, labor-saving, and repeatable. My background bridges architecture and technology, so I can take research and turn it into practical ways for building better. Earth might not replace concrete everywhere, but where it can, it matters, and it can change the picture. Thank you.
Have you made any experiments with this, with the results?
Yes, I did. We succeeded to print 3D print stimulated soil, but we didn't succeed in improve its properties yet, because we need to do more experiments to find the balance between stimulating the soil and use this solution to improve its properties, than the need for buildability for 3D printing. But it's the first year of the research, so thank you.
I'm standing here to keep the rules. So yes, but it's a great chance now. We have a break, so you can, of course, keep on asking the presenters more in detail if something is unclear or you would like to know more.